Log on, tune in, find out. Another good idea from Cambridge.
In the afternoon session, as you can see, Gary Rosner is the first speaker, and he's going to be speaking on Bayesian enrichment strategies for randomized discontinuation trials.
Thank you very much, and thank you, Sergey and Basi, for inviting me to this very interesting and great week. And so let me get started. So very quickly, I'm going to go over some of the issues that come up in Phase 2 study designs for oncology. Phase 2 study designs are changing right now in oncology because the types of treatments, pharmacologic treatments that we're developing in oncology are changing. And I'll talk a little bit about that. Then I'll talk about the use of decision theoretic considerations in designing studies. And I'll apply that in a particular type of study design called the randomized discontinuation design, and how we've incorporated a decision theoretic, how we've incorporated a tumor growth model within the context of the decision theoretic model that requires one to be able to make predictive statements about what's going to happen in future patients, apply a utility function, and come up with optimal designs. This is joint work with Lorenzo Tripa, who's now at Harvard, and Peter Mueller, who is now at University of Texas, Austin, except right now he's sitting right in the second row. All right, in Phase 2, the principal objective, at least in oncology, is to determine whether or not a drug has sufficient activity, where activity is defined in terms of some effect on the disease, obviously, enough activity to warrant further investigation. That further investigation may be in subsequent Phase 2 studies or maybe in a larger randomized controlled Phase 3 study. The goal tends to be what's called explanatory rather than pragmatic. That is, we're looking for some of the biologic activity and not necessarily trying to evaluate how the drug will fare in the real world where people don't always adhere to their treatment plans, etc. So clinical efficacy, that is how, if a drug actually works better in the real world, that would suggest that there is in fact activity. But just showing that a drug has a biologic effect doesn't necessarily mean that it's going to be a better form of therapy in the real world. So the objective in Phase 2 is more in terms of looking at activity. In general, in Phase 2, we want to have a quick answer. Many studies in oncology, in cancer, will, at least the Phase 3 studies, will be looking for survival-related endpoints. And for several different types of cancer, that could take years to get an answer. So in Phase 2, we want to find some kind of measure that hopefully will correlate with the ultimately important survival endpoint, but will give us a signal early on. We tend to have smaller studies requiring fewer patients, and as I mentioned, we want to have shorter studies so we have shorter follow-up. So the traditional designs that have been used in cancer and evaluating cancer drugs, those designs have typically looked at response versus non-response. Response would be measured in terms of tumor shrinkage. Does the tumor shrink by at least 50%? That would be considered perhaps a response. The other aspect of oncology drugs, at least historically, is that the more drug you give, the more you'll shrink the tumor, but also the more you give, the greater the toxicity. So you have to balance toxicity and efficacy. Newer agents are now targeting particular biochemical pathways. We've learned a lot about how cancers develop, how they grow, how they send out particular signals to get the body to produce new blood vessels, to provide them with nutrition, and so on. And so as we learn more about that, we're devising new forms of therapy that can inhibit or somehow trick the body either to not recognize it or recognize these signals coming from the tumors. So these targeted agents may not work the same way as the old ones that were more poisonous. Instead, they may slow or even just stop tumor growth for over some period of time. So it's tumor shrinkage by itself may not be the appropriate endpoint. Instead, stable disease, tumors that don't appear to grow quite so quickly, that may be an important endpoint. So the old study designs aren't going to work as well. So what are the things that we need in these phase 2 studies? We want to have good accrual, that is, patients will want to participate. We want to be able to determine whether or not a drug is active. We want to avoid biases caused by heterogeneity in the population in tumor growth rates. For certain types of cancers, there's great heterogeneity where some patients can appear stable over a long period of time just because of the natural characteristics of their tumor, whereas other patients may progress more quickly. So we would want to have randomization so that we can see whether or not our drug is having an effect, as opposed to just chance bringing in patients who tend to have slower growing tumors. And we obviously also want to be able to monitor how things are going so we can stop the study early if we want. So that's a little bit about the background for phase 2 studies in cancer. Now, what about decision theory and design? We've heard a little bit about optimal design or simulation-based clinical trials. In our case, though, we're going to use a utility function that's going to balance costs and benefits, and we're going to figure out an optimal trial design that will maximize the utility. So what are some of the decisions that are involved in designing a clinical trial? Well, we have to choose a sample size. We have to figure out how long we're going to follow the patients before we can make an assessment, or before we do make the assessment of whether or not the tumor growth is increasing, stable, or the tumor itself is shrinking. We need to include stopping rules during the study so that if it appears as though the study is not going to be fruitful, we want to stop early, or if it appears as though the treatment is having a big effect, we might want to start the phase 3 study sooner. And obviously an important decision, as I think Peter said this morning, is the optimal sample size 0? Should we even do this study? And why not make these decisions that go on implicitly in study design, why not make them explicit and incorporate them in some subset of these considerations into the utility function? So as we all know, in decision theory, we have a set of possible actions. So it might be sampling times for pharmacokinetics or for viral load. It may be numbers of patients. It could be follow-up times. So there are a set of possible actions that will define our decision space. Set of possible outcomes, how the patients will respond to the treatment. And then we'll have the parameters that characterize our underlying response. And these will be stochastic in nature. And then we will put on top of that a utility function that will relate the outcomes and the actions to ultimately the utility. And we'll want to pick that design that's going to maximize the utility. We take a Bayesian approach, which basically says that everything is stochastic in nature. We have more or less certainty about certain states of nature, and we're going to use that uncertainty, acknowledge it, and incorporate it in our decisions, in our decision making. So we have uncertainty in terms of the parameters in our model, and there's naturally the variation that occurs in patient outcomes, person-to-person variation, and we're going to incorporate all those things and integrate over all the sources of uncertainty to get our utility for a given action. Then we pick the action that maximizes the utility. A simple example would be in a pharmacokinetic setting where we might have, in this case, this would be the concentration time curve. The systemic exposure for this patient may be too short with respect to some pre-specified target of area under the curve systemic exposure that we think is the best to achieve the outcome we want. This patient may be too high, and what we would want to do is we would want to take all the uncertainty for the next patient and apply it with respect to a utility function and figure out what's the best dose to get their AUC within that target range. So in this case, our utility function has the outcome, the design, which would be, in this case, the dose that we give, and then the parameters associated with the pharmacokinetic model. So we have a loss function that's a function of the area under the curve. AUC would be a function of those parameters and the dose. We're going to pick the dose that's going to maximize, that's going to, in this case, minimize our loss. So our utility function here is a loss associated with being outside that range. And we want to pick that dose to maximize that. So as an example, we could have a loss function that's represented here. The x-axis is the area under the curve, and the y-axis would be the loss. And so we have an asymmetric loss in this particular example. So if you're outside this target range by being too low, then you incur a loss, but not quite as steeply as being above the AUC, considering the fact that if you're above the systemic exposure by too much, you may kill the patient. So the loss function here, these slopes are not equal. But we can characterize or come up with some sort of loss function working with our clinical colleagues. For a given patient, we might have historical information from previous studies of the relationship between two parameters in, say, a one-compartment model. In this case, we have volume and the elimination rate constants. So we might have different studies that give us some idea of the distribution of these two parameters, the joint distribution across the population. We might have some information for a given patient, the next patient that we're going to treat, and then we can incorporate that patient's uncertainty with the loss function, integrate that, and determine for that patient that this dose would be the optimal one in that it minimizes the loss. It's the one that's going to be the most likely to get the patient within the target AUC. So that's a brief idea of how decision theory could be applied in designing a pharmacokinetic study. A study that you want to maximize the probability of the patient exposure to the drug, minimizing the loss. So as I mentioned before, in cancer now, rather than having cytotoxic therapies, we might actually be developing cytostatic therapies, drugs that are going to stabilize tumor growth rather than cause an immediate shrinkage. And so stable disease itself may be a goal, at least over some reasonable period of time. Again, it's a different way of thinking, and so in some respects, stable disease might be considered as a problematic endpoint, depending upon how long the follow-up is, but it may well be a useful endpoint. As I mentioned, we need randomization in our study designs because many different cancers have great heterogeneity in the patient population in terms of tumor growth. We want to be able to eliminate the possibility that chance has given us a large number of patients with slow-growing tumors as opposed to the treatment having the effect. And we're interested in activity. And we may have to enroll more patients than we did in the past, as you'll see. So I mentioned all this already, that the newer drugs are targeting particular biochemical pathways that may be involved with the initiation or the what's called angiogenesis, that is the development of new blood vessels that are feeding tumors and allow the tumors to grow, or different pathways that allow a primary tumor cells to come off of that primary tumor and move through the body to form metastatic disease. How best can we measure the pharmacodynamic effect? Is another issue, whether we use radiographic measures, whether we use biopsies. I won't be talking about that in this talk, but that's also an important characterization and something else that might be incorporated as part of the assessment of the outcome. So one particular approach to evaluating these newer designs would be to use an enrichment design. And in cancer, or in several other diseases as well, people are now starting to consider enrichment designs from Bayesian standpoints as well as from non-Bayesian standpoints. you start a study with a large group of patients, but as you carry out the study, you want to learn which subpopulations are the ones who are truly benefiting, or perhaps benefiting the most. And then you might adaptively change the enrollment criteria so that you enrich the population in your study for those patients who are truly benefiting. Some proposals include randomized discontinuation designs, like the design I'll be talking about, and we propose this in Journal of Clinical Oncology. There's also a cross-validated adaptive signature design for cancer studies that's been proposed by Rich Simon and Boris Friedland. So let's talk a little bit about randomized discontinuation. So as I mentioned, the challenges for phase 2 studies of these newer agents are the heterogeneity in tumor growth rates, and that perhaps only a subset of the population may benefit. Might be patients who have a particular mutation in their tumors, and so they respond more to the particular drug than patients lacking that mutation. Or in fact, in some situations, patients who don't have mutations, who have the wild type cancer, are the ones who benefit. So we're going to talk about the randomized discontinuation design. It goes back to a paper by Kopek et al. From the Journal of Clinical Epidemiology in 1993, where they proposed this design. Historically, the first mention of it was actually in the Journal of Clinical Pharmacology in 1975, and they claim that the randomized discontinuation trial design has certain advantages. The way they applied it, basically each patient would be titrated to his or her own best dose, and then they would be randomized either to continue on that best dose or to whatever the control treatment is. So you're kind of stacking the deck so that for each patient, they would have the highest chance of benefiting from the drug. That's not the way that we're going to apply it, though. But in general, there are two phases for the drug. So it could be an open label phase where you do the dose titration, or in our case, everybody's going to get the same dose of the drug. And we're going to find the best dose, or we might just remove patients who don't adhere to the drug, either because of toxicity or something, or other reasons that will kind of enrich the population by the end of this first phase. So patients will continue on to the second phase if they seem to be responding, or if they're able to tolerate the drug. And then we'll randomize the successful drug takers or the responders in the second phase to either continue on the drug or to go to the control treatment, and there'll be a double-blind randomized clinical trial at that second phase. Schematically, there's the open label phase wherein the patients receive the drug for a particular length of time to be determined. So part of the optimal design that we'll talk about is how long should we wait until we make this assessment of whether or not it's okay to start randomizing patients. Did the patient respond to the drug? Is the patient tolerating the drug? Is the patient taking the drug? If not, then they're out of the study. This is a cancer study. If the patient's tumors actually seem to be shrinking, then they continue on the drug. But if they're in this kind of vague zone where they may be benefiting or they may just have slow-growing tumors, we randomize them either to continue the drug or to go on to placebo. I just note here that patients who are randomized to placebo, if their disease happens to deteriorate, it progresses, then they can switch back to the active drug. So this type of design is useful when we're interested in non-curative therapies, perhaps, okay, but that things can, the patient can be steady for a relatively long term. There may be a small treatment effect, certainly in the larger population, so we enrich to maximize our signal relative to the noise. We want to minimize placebo use, so rather than upfront randomizing everybody, say, to placebo versus active drug, everybody gets the active drug at first. And we want to minimize the effect of heterogeneity. We do that through the randomization. And we're not really estimating the size of the treatment effect in the general population. We're not estimating effectiveness, but we're interested in activity. So explanatory as opposed to a pragmatic study. And I know Val did some work early on about comparing randomized discontinuation design and show that there are different situations where this design can improve over just a standard randomized design. So what are some limitations? Well, we may need to have many patients whom we enter into the study if it's a negative phase 2. We're assuming that there's no carryover effect because after the randomization, some of the patients are going to stop that active treatment. And so we're assuming that there will be no continued benefit, at least over the majority of the period of follow-up following randomization. The analysis gets complicated if patients drop out or refuse randomization. And it may be difficult to identify up front which patients are the ones who are most likely to go on to randomization. But that's obviously an open issue to consider. So we actually implemented this when I was with the Cancer and Leukemia Group B, which is one of the large consortia of different academic cancer centers in the United States, funded by the National Cancer Institute. We carried this out in a study of metastatic kidney cancer, which tends to have associated with it a variability in the growth rates across the population. And so we might actually see some patients whose disease is stable in the absence of therapy for 6, 12 months, or even more. So that heterogeneity is something we obviously want to control for, hence we want randomization. And so we proposed and actually carried out a randomized discontinuation design. By having this sort of open label phase where everybody gets the new drug, it turns out that we can have very fast accrual to the study. Patients prefer that. And also, patients are told that they can continue the therapy if they're benefiting. Stable disease was considered a valid endpoint over in this particular setting. And we incorporated randomization, as I mentioned. And we also had early stopping rules, though I won't talk about those here. So this was the schema for that study. They got a drug that was supposed to be an anti-angiogenic, that is, it would stop the creation of new blood vessels to the tumor. We would evaluate them after 16 weeks to see whether or not their disease is stable. If they've progressed at any time during those 16 weeks, they come off the study. If they actually had some tumor shrinkage over those 16 weeks, they stay on the drug. And if they were stable, then they get randomized to continue the drug, being followed for another 16 weeks, or to go to placebo, again, evaluation after 16 weeks. So the primary analysis was to compare the drug CAI to placebo after 16 weeks post-randomization, and the primary endpoint was just the fraction of patients who progressed over that second 16-week period. And the information was percent of patients who were stable after the first 16 weeks, the open label phase, and then there's the randomized part after that. Turned out the drug didn't work, and so it was we actually stopped the study early, and it was a negative study. So now what we'd like to do is, how can we improve on that design by using decision theory? So we're going to apply decision theory to help choose certain design parameters. How long should that open label phase be, during which everybody gets the drug, and at the end of which we evaluate whether or not the disease is stable and they should be randomized? How long should the follow-up be after randomization? Again, these are both characteristics of the tumor growth, which we're going to estimate using historical information, and of course the number of patients to enroll. So those are going to be the three key characteristics that we're going to evaluate, or we're going to pick as our design points. And it's all a function of all of these things. And this is all based on a paper that will appear in Biometrics with discussion by a few people. So the particular growth model that we use is this Gompertzian tumor growth model. Use of a Gompertz model to characterize tumor growth actually has a long history. Whoa, wrong button. we allowed each patient to have his or her own parameters here. So there was and with a distribution on those across the population. So we have a stochastic differential equation. And this just talks about the tumor size at time t for the ith patient. And it's a function of again, this is the Gompertzian model. But we also have this Wiener process here to characterize variation over time. And then there's also, because of the fact that we have between subject variability in these parameters, we also have between subject heterogeneity in the tumor growth rates. We have historical data, we're assuming at least, we have historical data at discrete times of tumor measurements for a group of patients. And we're going to use these prior growth curves, fit that model so that we can estimate something about those, about the population model for the parameters in this Gompertzian model. And in fact, we're also going to use the historical information in a somewhat novel way as our prior for the new study that we're going to design. So we treat these historical values at discrete times as fixed, and we're going to use the Gompertzian model to actually just interpolate between observed times for a given patient. We're going to use the Markov structure, which is on the next slide, so that we can use the model and figure out a tumor growth, a tumor size at any point in time given the previous time. And we're using this empirical model based upon the observed data, the historical data, for prediction of future patients. So we combine those as well as allowing the fact that these parameters may change at the time of randomization. So it would be piecewise in that before randomization, when everybody's on active treatment, they have particular parameters. And then after randomization, if they go on to placebo, those parameters would change. And we can predict future tumor growth, as I mentioned, through this, the Markov structure that's inherent in the model, where we can actually write out what the probability or the probability distribution or the sampling distribution of future tumor sizes given a current tumor size and the parameters. So we'll need to be able to generate future patient tumor growth curves in order to make predictions, because we're going to require the predictive distribution in order to optimize our utility function. So we do that again via the three-parameter Gompertz diffusion process I mentioned with a hierarchical model on the subject-specific parameters. We have interpolation to any time so that we can characterize a continuous curve based on just those discrete points using the Markov structure. We can easily construct and actually demonstrate for our colleagues, our clinical colleagues, what the tumor growth curves would look like, which are easily interpreted. And then we also reparameterize things so that there's just a single treatment effect parameter in the model. And then we can elicit from the experts what they think the treatment effect might be and how likely it would be this large or that large. So again, we have this random sample of historical measurements at discrete times. We've again used this interpolation. The interpolation, also the fact that it's anchored to observed times makes it, and we're only interpolating between those times, makes this model more robust than, given that it's purely empirical, than another model might be, like assuming it's log linear or whatever. So if our model assumption may not hold over the entire time course that we're looking at, but between these discrete times of tumor measurement, it should be fairly good. And we can elicit the treatment effect from our colleagues and show resulting predictions. That's an important part of how we design studies using Bayesian methods in general, by sitting down with the colleagues and our clinical colleagues and saying, if you make this assumption, this is what you're going to see, that assumption, this is what you're going to see, and go from there in terms of the iterative process of designing the study. So for example, over here we have at these points, so there's a box there and one there and one there. Those are the actual measured tumor sizes in a particular clinical study. Actually, it was that CAI study I mentioned before. These squiggly lines that go through those points are just realizations of that stochastic process. In this case, it's in the control setting, or for placebo. This would be an assumed treatment effect. So this is how the treatment, if you assume a particular size of treatment effect, how it might affect tumor size, tumor growth over time. We can then also project trajectories for general patients. We have an expected value, and then we also have the alternative values for different treatment sizes. So we can use these. This is how we're going to use them in designing our study. So once again, we're going to simulate the different studies, and it's important to show how these assumed treatment effects change the operating characteristics of the study. So we'll do that by also simulating a bunch of clinical trials and documenting for different trial designs what would be the important characteristics that perhaps would be easier for the clinician to understand rather than just looking at tumor curves. What's important is how frequently are we going to have a successful study, not successful, how large a sample size we're going to need, and so on. So we can summarize that by three probabilities. The probability that a patient becomes eligible for randomization after the open label phase, and then the response probabilities for treatment 0, say placebo, and treatment 1, the active drug, after randomization. And we can produce tables for these values as they're implied by different treatment effects in terms of tumor growth, and show that, sit down with our colleagues and go over that. All right, and I'll skip that because I've been saying that. So now we want to formulate this as a decision problem. We have a distribution of data based upon the historical information that will allow us to predict future growth, tumor growth in future patients. We have a distribution of model parameters, again, coming from historical information. And we have an action space of alternative designs. Those alternative designs would consist of how many patients we enroll, how long we follow patients during the open label phase, and how long we follow them during the post-randomization phase. So we have a utility function that's going to consist of a cost and a benefit. And we want to balance the cost versus the benefit. So the cost would be a function in this case of how many patients we enroll and the cost of those patients, of enrolling them, how many patients we follow in the post-randomization phase, and the cost associated with treating them in that second phase, and then an overall cost per unit time for the number of patients we enroll whom we follow for the first open label phase, that would be for time T1, and then the ones that we follow, the follow-up cost for the ones after randomization, small n, and post-randomization time is T2. So the key design parameters are n, the number we're going to enroll, the duration of follow-up during the open label phase, and the duration of follow-up during the post-randomization phase. The benefits we're going to parameterize or characterize as a benefit arising from a subsequent randomized phase 3 study that shows that's a positive study and shows a treatment effect. So it has an indicator function that in this case it'll be a randomized phase 3 study with typical frequentist type design, and the probability or an indicator function that the test statistic is in the rejection region, and that's going to be scaled by an expected treatment effect, so that the larger the treatment effect, the greater the benefit. So it's not just that the subsequent phase 3 study that we predict is going to be significant, but also the magnitude of the treatment difference. And so our utility function just balances the benefits against the costs. So how do we implement that? We're going to simulate the trial for various values of these three parameters: the number we enroll, the duration of follow-up in the pre-randomization period, and the duration of follow-up after randomization. The number who actually get randomized, little n, is a function of T1 and the distribution of tumor growths across the population. So we don't have to put that in there as a design parameter. And a patient will continue if their disease, at the time of evaluation, if the size of the tumor is between some boundary, so that it's considered stable. We use Monte Carlo to optimize the utility function, and I'll show you how to do that in a second. But we're going to approximate the expected utility function by simulating the trial for various values of these three parameters, and then just fitting a surface across that, so that we not only average for a given configuration of these three values, these three design parameters, but we also smooth, so borrow some strength from the utilities associated with neighboring values. And as an example, we're just, in this case, simulating these two underlying growth trajectories. This is for placebo, this is for the treatment, and the solid lines on the, this is the median, and these are the, I think, 90th percentiles. The dashed lines just correspond to the same percentiles, but for the mean. So that's placebo, and that's for treatment. We then carry it out on a grid of different, we just do a random sample on this grid of different values of n, T1, and T2. And we get these surfaces. I can only show projections. I can try to show 3 dimensions, but I can't show 4. So these are basically slices, 3-dimensional slices of the utility surface. And these would be just a single planar view of the utility as a function of the sample size over here. And we can find here the maximum or optimal sample size. We can find the optimal duration of follow-up pre-randomization and the optimal duration of follow-up post-randomization just based on these smooth surfaces. And some example solutions over here. This would be taking the historical information from that CAI, the Cancer and Leukemia Group B study that I mentioned early on, told you about. We have tumor measurements from that study. We use those with this Gompertz model and simulated a particular, a new study, a future study, to pick the optimal design using using various values of costs. And so one thing to note is that when we have, let me go back, when we're looking at T1 over here, the time of the open label phase before randomization, if T1 is equal to 0, that would correspond to randomization from the start. So if there's no open label phase, but there is a subsequent to randomization duration of follow-up, that means we randomize it up front. So implicitly or explicitly, this includes a comparison of the randomized discontinuation design to upfront randomization. And in this case, the decision maker will favor a randomized discontinuation design over just a randomized upfront 2-arm design, and also over no study where n is equal to 0. What we would do then is, for particular values, we can also produce a table of operating characteristics, as I mentioned before. So if we assume roughly 12% tumor size for the treatment and 24% for the control, the optimal design has certain values in terms of the number of patients you'd have to enroll. You'd have to follow them for 72 days before randomization, 145 days after randomization, based on what was seen in that earlier study. And we can produce tables like this for certain proportions of patients who would go on to randomization from 0.3, say, to 0.8, and efficacy being low, a low treatment effect, a medium treatment effect, or a high treatment effect, where those have certain values with respect to how the treatment works. We can produce probabilities of rejecting no treatment effect, so something like power, under different susceptibility probabilities, these pi's. So the more patients who go on to randomization with high treatment effect, well, you can see as more patients go on to randomization, we have higher power, which obviously makes sense, but you can also compare that to what you think the treatment effect might be. So in summary, the randomized discontinuation design was feasible. It's been used several different studies in oncology. One can apply decision theoretic considerations to design the clinical studies. In particular, we can come up with a utility function that captures competing needs. We can base the complex repeated measures of tumor growth on historical data using the stochastic differential equation model. Simulation-based optimization is a useful method where you just simulate across this grid of design points and then fit a surface and find the optimal that way. And then it's important also to evaluate your study with respect to the operating characteristics. Thank you very much.
Question? Yes, Nancy.
First, a comment anyway. When one was just looking at the unimodal toxicity function and trying to estimate a target, the optimal design for doing that was to put all your points at the unknown target. And so there was at least some logic to trying to place your subjects at that point. For the toxicity efficacy situation, if you have a curve that in some sense goes up and goes down, for all of the models that I know of that have been evaluated, there are no one-point optimal designs. If you want to know the maximum of your utility function, the optimal design will be at least a two-point design, and none of the points will be at the maximum. So what I'm questioning, and I don't know whether you've thought about it or not, is this strategy of predicting the maximum and then putting the next patient at that point, because it's not optimal. And I don't know whether you've thought about doing something else.
So you mean making this into a sequential type design? So as you
Well, it is
recruit
a
some
sequential
patients,
design.
not in the sense that we're changing the design characteristics, that is the duration of follow-up for the pre and post. That's not changing once the study starts.
You're selecting the dose to give the patient. You have a treatment allocation rule, which is to put the patient at the dose that maximizes your predicted utility.
Dose didn't enter into this at all. There was a fixed dose that was used.
Well, there was a fixed dose. So your utility function,
Is my utility...
Can you show your algorithm again? You're putting the
You're talking about this utility function you wanted to see?
No, the one where you say where you're the, maybe I mixed up something here.
Show the
What is this slide?
plot with utility function. It was some kind of utility. No.
Not that
Before that, do you remember it was some kind
you
of
see.
bucket?
Keep going
shape?
and I'll see.
Oh, that one.
No.
That example from the AUCs? Yeah, that was just an example of the use of utility function in a particular pharmacokinetic study where you wanted to maximize the
So you're
probability
talking
of
about a
patient.
different study. and
That
then
was just an
Okay,
example
well, I got
application.
up there.
Sure, sorry. Steve, oh,
A couple of questions about the use of stochastic differential equations, and it's good you have this slide. First of all, have you analyzed the properties of the solution of the stochastic differential equation here? Why I'm asking, there were a number of publications on the use of stochastic differential equations in pharmaceutical applications, where in fact the model, what happens, you can prove that the probability that the trajectories are negative is positive, and sometimes it's exactly 1/2. So when you add this term to the Wiener noise, when you multiply it by the state, it goes away. But we have not analyzed the model which you have. You have this x log x. I'm just wondering whether you have looked at this or not.
so we got this from the literature, and I honestly don't remember whether or not they talk about that in the paper.
Okay, divided by x post part, and it will be equation about log. So
You have to be more
it is
careful
not-
with
Yeah,
stochastic
but it's
differential
no
equation.
problem here, because it can be reduced to differential equation with respect to log of x. And while we are on this slide, you see, you applied to stochastic differential equation to tumor growth. And this process known that it's rather smooth process. And any solution for, say, this stochastic differential equation, it will be really not very smooth. It will be continuous, but derivatives can be infinite. So instead of that, and by the way, mathematically it could be simpler, instead of white noise equation, it's better to add some kind of, you see, what they call in engineering color noise. When you have, instead of a Wiener process, you have actually piece of expansion of Wiener process with respect to some orthogonal function. And then, Peter, you look at me, so probably you're somehow in this business more involved. So then it's easier to just to include a few terms which people do in engineering, so-called columns. And then, first of all, your trajectory will be smooth. Even derivative will be smooth. And I think it's simpler mathematically, and it feeds to at least my conception of tumor growth. So the first question, and while I'm still standing, it's difficult to stand up. It's easy to sit down. And you have also some kind of, you assume that you classify your patients very well. And because it's, you see, it's dynamic process, it could be false classification, it could be a lot of false classification. And if you really look at how they measure all these tumors and lesions and whatever, so you are in trouble. So false classification is a problem there. So did you try to address it?
No, we didn't. But I did have, on one of the slides, I mentioned how do we measure response. That I'm not going to talk about that in this talk, but that is an important consideration now. Is that that's what you mean?
Yeah.
Yeah, no, there are a lot of problems with that.
Phases in data analysis, final data analysis. I mean, phase 2 and phase, I mean, before splitting patients to groups. So did you use this information from the very first stage in the final analysis? Or you use only, for final analysis, you use only information from the final stage, the final stage?
So in this particular thing, in our utility function, it enters into the predictive distribution for the subsequent phase 3. So that's looking at the treatment effect, the posterior distribution of the treatment effect, which in essence comes from, I guess, just the post-randomization part, because that's where you compare the placebo control to the treatment. But we're estimating the treatment effect itself throughout the entire course that the patient is on the treatment.
Okay, so you assume that when you do your Monte Carlo, you assume that at the end you do analysis. You do not do actual data analysis, I understand. But when you compute your
right, but we're not right. But unlike the earlier randomized discontinuation design, the CALGB study, unlike that one where I was maybe projecting to the end of that study, in this one we're projecting to a future phase 3 study.
Okay, thank you.
Okay, thank you, Gary. I know there's other questions, but we're already eating into Peter's time. So Peter's already been mentioned, and